Welcome back, everyone, to this lecture in general biology. Today we'll be covering the 9th chapter of Campbell biology, cellular respiration, and fermentation. Life is work. Living cells require energy from outside sources to do work. The work of the cell includes assembling polymers, membrane transport, moving and reproducing. Animals can obtain energy to do this work by feeding on other animals or photosynthetic organisms, such in the case of this poppin. Energy flows into an ecosystem as sunlight and leaves as heat. The chemical elements essential to life are recycled. Photosynthesis generates molecular oxygen and organic molecules, which are used in cellular respiration. Cells use chemical energy stored in organic molecules to generate ATP, which powers biological work. Catabolic pathways release stored energy by breaking down complex molecules. Electron transfer plays a major role in these pathways. These processes are central to cellular respiration. The breakdown of organic molecules is exergonic. Fermentation is a partial degradation of sugars that occurs without the presence of oxygen. Aerobic respiration occurs or consumes organic molecules, such as molecular oxygen, and yields ATP. Anaerobic respiration is similar to aerobic respiration, but consumes compounds other than molecular oxygen, and usually results in a far lower yield in ATP. Cellular respiration includes both types of respiration, but is often used to refer to aerobic respiration in the presence of oxygen. Although carbohydrates, fats, and proteins are all consumed as fuel, it is helpful to trace cellular respiration with the sugar glucose, where glucose reacts with 6 molecular oxygens to yield 6 carbon dioxides, 6 water molecules, along with the release of energy that can be used to make ATP and excess heat. For redox reactions, those are a combination of oxidation and reduction. The transfer of electrons during chemical reactions releases energy stored in organic molecules. This released energy is ultimately used to synthesize ATP. Oxidation usually involves, in a typical general chemical way, a rise in positive charge or a reduction in negative charge, whereas reduction is a increase in negative charge or a rise in or a decrease in positive charge. And in an organic chemical sense, oxidation is the increase in bonds to oxygen and decrease in bonds to hydrogen, where reduction is the increase in bonds to hydrogen and lowering of bonds to oxygen. Redox is both occurring at the same time. The principle of redox. Chemical reactions that transfer electrons between reactants are called oxidation-reduction reactions, or redox reactions. Looking at just the general chemical side, in oxidation, a substance loses electrons or is oxidized. In reduction, a substance gains electrons or is reduced. The amount of positive charge can be thought of as reduced. For example, taking solid sodium and chlorine, the sodium becomes positive after reacting, therefore it becomes oxidized or loses an electron. Chlorine gains electron, therefore it becomes reduced or gains more negative charge. An example can be just oxidation, where negative charge decreases after a chemical reaction, and a reduction can be where negative charge increases. This is an example of just a simple electron being transferred. The electron donor is called the reducing agent. The electron receptor or acceptor is called the oxidizing agent. The oxidizing agent gets reduced. The reducing agent gets oxidized. Some redox reactions do not transfer electrons, but change the electron sharing in covalent bonds. An example being a reaction between methane and dioxygen. What we're seeing is just some Lewis electron pairs. And simply put, what we have is a reordering of how those electrons are shaped, where methane has 4 sets of lone pairs between carbon and separate hydrogens, and then when carbon gets transferred into carbon dioxide, it gains 2 sets of double bonds between 2 different oxygens, whereas the starting oxidizing agent O2 becomes reduced to H2O. So again, things that add more bonds to oxygen is the oxidation process, and things that add more bonds to hydrogen is the reduction process. Or half reactions. During cellular respiration, the fuel, which in this case is glucose, is oxidized and dioxygen is reduced. Organic molecules with an abundance of hydrogen are excellent sources of high energy electrons. Energy is released as the electrons associated with the hydrogen ions are transferred to oxygen, a lower energy state. Stepwise energy harvest via NAD+, and the electron transport chain. In cellular respiration, glucose and other organic molecules are broken down in a series of steps. Electrons from organic compounds are usually first transferred to NAD+, which is a coenzyme. As an electron acceptor, NAD+ functions as an oxidizing agent during cellular respiration, becoming NADH, the reduced form of NAD+. This represents stored energy that is tapped to synthesize ATP later. What we have is nicotinamide, or NAD, is oxidized form, NAD+, and then becomes its reduced form, nicotinamide, without the positive charge, but the gain of an additional hydrogen. Or a hydride, negative charge. Further looking at the chemical reaction that occurs between nicotinamide in its oxidized positive form and its reduced neutral form. What we're looking at is dehydrogenase, which assists in oxidizing what appears to be an alcohol to a carbonyl, a C double bond O, thus reducing nicotinamide to its reduced form with the extra hydrogen and releasing a proton or a H+ ion. Reduced nicotinamide passes the electrons to the electron transport chain. Unlike an uncontrolled reaction, the electron transport chain passes electrons in a series of steps instead of one explosive reaction. Dioxygen pulls electrons down the chain in an energy-yielding tumble. The energy is used to generate a ton of ATP. In an uncontrolled reaction, this could release a lot of energy, which would be hard to capture a good amount of it, whereas doing stepwise controlled steps in the electron transport chain, energy is slowly released or pulled from that high energy electron in a variety of steps before dioxygen accepts the energetic electron along with some originally released protons to form a stable water molecule. As a preview of the steps, harvesting energy from glucose happens in 3 core stages. There's glycolysis, the citric acid cycle, and oxidative phosphorylation. Glycolysis, as the term lysis implies, is the breakdown of glucose into two molecules of pyruvate. The citric acid cycle completes the breakdown of glucose by occurring a full cycle per pyruvate molecule produced in glycolysis, and then after the end products are released, oxidative phosphorylation accounts for most of the ATP synthesis. All right. Initially, glucose gets broken down to pyruvate as electrons are provided from the reduced nicotinamide. And then some ATP is produced during this short process, but not much. As this enters from the cytoplasm into the mitochondria, the pyruvate can be oxidized into acetyl coenzyme A. And through the citric acid cycle, plenty of coenzymes are produced, such as the reduced form of NADH and the reduced form of FADH2, which again produces a little bit of ATP. And then oxidative phosphorylation, the electron transport chain and chemiosmosis, produces a large amount of adenosine triphosphate from released electrons made available through NADH and FADH2. The process that generates almost 90% of the ATP from a glucose molecule is called oxidative phosphorylation because it is powered by redox reactions. A smaller amount of ATP is formed in glycolysis in the citric acid cycle by substrate level phosphorylation. But the key of how oxygen comes into play is through the electron transport chain, which is why we are able to produce a lot more ATP per glucose molecule than most unicellular life. So what we're looking at is an enzyme capturing inorganic phosphate and adenosine diphosphate to form a product, and ATP, or I should say, the starting reactant was a phosphorylated substrate, and then the phosphate group moves to ATP. For each molecule of glucose degraded to carbon dioxide and water by respiration, the cell makes up to 32 molecules of ATP. There is a range depending on whether we are looking at it biochemically or through molecular biology. So you may see different numbers in the amount of ATP depending on which future course you eventually take. But here it's okay to know that up to 32 is usually within reason. We can use money as an analogy for cellular respiration. Glucose is like a larger denomination bill. It is worth a lot, but it is hard to spend at any given place, especially if places like McDonald's don't want you having more than a $50 bill. ATP is like a number of smaller denomination bills of equivalent value. They can be spent more easily, though you'll lose a little bit in tax along the way. Cellular respiration cashes in a large denomination of energy from glucose for a smaller change as numerous ATP molecules that can be more easily transported throughout the body. Glycolysis, sugar splitting, sort of like hydrolysis, splitting through water, breaks down glucose into two molecules of pyruvate. Glycolysis occurs in the cytoplasm. It has two major phases, the energy investment phase to get things started, and then the slight energy payoff phase at the end. Glycolysis occurs without oxygen. Therefore, this occurs in both aerobic and anaerobic respiration. Here we're seeing glycolysis begin, and then the next stages will occur later. Here we see 2 ATP are initially invested to get glycolysis started, and then 4 ATP are formed, causing a net production of 2, But also, very importantly, two reduced forms of nicotinamide are made as well, NADH, and some excess protons, H plus ions, are released, and all these come into factor later on down the cellular respiration chain. Here we're looking at some of the various different enzymes that are at play, starting with hexokinase, a kinase that phosphorylates a hexose sugar or a six-member sugar, six-carbon sugar. We go from glucose to glucose 6-phosphate, and phosphoglucoisomerase is a isomerase enzyme that changes not the chemical formula but the chemical structure. Therefore, it isomerizes glucose into fructose, or glucose 6-phosphate into fructose 6-phosphate. And then phosphofructokinase phosphorylates phosphofructose, or fructose 6-phosphate. And then we're left with aldolase, which creates glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are able to isomerize between each other through isomerase. Here we're looking at the initial energy investment. ATP needs to be invested to get glucose first started down this chain. As we continue on from the fructose isomerized version, that also needs to be doubly phosphorylated. And then as it gets broken up, we have two phosphorylated smaller molecules. Now, glyceraldehyde 3-phosphate, whether it's initially produced or it's isomerized from dihydroxyacetone phosphate, can go through triose phosphate dehydrogenase to produce two reduced forms of NADH, and then through phosphoglycerokinase we get the phosphorylation of two ADPs, so returning a net yield of 0 ATP so far. But continuing forward, phosphoglyceromutase mutates or rearranges the molecule a bit to 2-phosphoglycerate, and then enolase works on the molecule to form phosphoenol pyruvate. And then we're left with pyruvate kinase, which yields 2 new ATP for a net yield of 2 ATP through glycolysis, yielding a molecule of pyruvate. Just note that one molecule of glucose produces 2 molecules of pyruvate. Therefore, overall, 4 ATP are made from one glucose molecule so far, because we have 2 pyruvates. And looking at all this, in the presence of O2, pyruvate enters a mitochondrion in eukaryotic cells where the oxidation of glucose is completed. Before the citric acid cycle can begin, pyruvate must be converted to acetyl coenzyme A, which links glycolysis to the citric acid cycle. This step is carried out by a multi-enzyme complex that catalyzes 3 reactions: the oxidation of pyruvate to release carbon dioxide, the reduction of nicotinamide, and the combination of the remaining 2 carbon fragment molecule in coenzyme A to form acetyl coenzyme A, or acetyl-CoA. Now we have just finished. Now we are about to enter. We finished glycolysis and we're about to enter pyruvate oxidation. A lot of this is more stuff that would be highly remembered for a biochemistry class or a cellular molecular biology class, which I personally experienced in my entire junior year and the spring of my junior year, respectively. But this is general biology, so we're just going through the initial information that you might use later in your coursework. Going from the cytosol of a cell into the cytoplasm, what we see is that pyruvate gets transported, carbon dioxide is released. Also, I believe that the publisher has an error, usually a carboxylate group. I believe that O positive should be an O negative. All right. The publisher will need to be notified about that. Anyways, we reduce another form of nicotinamide, and coenzyme A lops onto the remaining two carbons from our original pyruvate, forming acetyl-CoA. And since we made two pyruvate molecules from one glucose, we at this point have two acetyl-CoAs. The citric acid cycle is also called the Krebs cycle, and in biochemistry is also called the tricarboxylic acid cycle, TCA. The citric acid cycle completes the breakdown of pyruvate into carbon dioxide. The cycle oxidizes organic fuel derived from pyruvate, generating 1 ATP, 3 NADH, and 1 FADH per turn of the citric acid cycle. Of which we'll have 2 turns from our 2 pyruvates from our 1 glucose molecule. The citric acid cycle has 8 steps, each catalyzed by a specific enzyme. The acetyl group of acetyl-CoA joins the cycle by combining with oxaloacetate, forming citrate. The next 7 steps decompose the citrate back to oxaloacetate, making the process a cycle. The NADH and FADH2 produced in this cycle relay electrons extracted from the food to the electron transport chain. And now we have the citric acid cycle. After pyruvate oxidation to form acetyl-CoA, essentially what we're looking at now is the citric acid cycle, which has various steps along the way. And while often many chemical reactions are reversible, it can only be reversible if we have the products in one place to go back to reactants. Something to note is that when a product is carbon dioxide, such as in part of the citric acid cycle, that carbon dioxide usually leaves, therefore it prevents the cycle from moving backwards at that point and is committed to move forward. All allowing us to reliably use this type of biochemical reactions to get multiple ATP and reduced coenzymes to help produce many more along the way. So as acetyl-CoA binds with oxaloacetate, citrate is formed, and then we get isocitrate. A reduction of nicotinamide leads into alpha-ketoglutarate, and then more reduced nicotinamide leads to succinyl-CoA. Then we have not ADP, but guanine diphosphate, which gets phosphorylated into guanine triphosphate, turning succinyl-CoA into succinate. Now, guanine triphosphate can be used later on to phosphorylate ADP to adenosine triphosphate. Fascinating how these molecules that look similar to our DNA monomers can actually be used in different ways. I wonder if there is cysteine triphosphate and thiamine or uracil triphosphate, but guanine and adenosine seem to make sense. They are both purines. I believe. Succinate then later can be used to reduce fat H to become fumarate, and then fumarate through the introduction of water becomes malate. And this allows further reduction of NADH to re-establish oxaloacetate. So just a closer up look of that chart that we just went through. Looking good. Looking good. Following glycolysis, the citric acid cycle, NADH and FADH2 account for most of the energy extracted from food. These two electron carriers donate electrons to the electron transport chain, which powers ATP synthesis via oxidative phosphorylation. The electron transport chain occurs in the inner membrane of the cristae of the mitochondrion. Most of the chain's components are proteins, which exist in multi-protein complexes. Electrons drop in free energy as they go down the chain and are finally passed to dioxygen, which combines with protons or hydrogen ions to form water. Electron carriers alternate between reduced and oxidized states as they accept and donate electrons. Electrons are transferred from NADH and FADH2 to the electron transport chain. Electrons are passed through a number of proteins, including cytochromes, each with an iron atom, as they transfer to dioxygen. The electron transport chain generates no ATP directly. However, it breaks the large free energy drop from food to dioxygen into smaller steps that releases energy in manageable amounts. So now we're going to look at that final process, the oxidative phosphorylation, that releases the majority of ATP. Here we're looking at a very huge complex, 1 through 4, each consisting of multiple proteins with electron carriers. What's relevant is that NADH and FADH2 that were produced later are now getting oxidized, which is allowing the course of this electron to go through multiple steps to eventually reach water. And this is a bit complex for general biology, so we'll leave it at this visual. But electrons are basically being released from being stored in our reduced coenzymes that are then being transported down this chain at certain parts. Eventually being accepted by dioxygen, binding with 2 protons to form water. The energy released as electrons are passed down the electron transport chain is used to pump hydrogen ions from the mitochondrial matrix to the intermembrane space. Protons, or H+ ions, then move down the concentration gradient back across the membrane, passing through the protein complex ATP synthase, something that's used to synthesize ATP. As these hydrogen ions move into binding sites on the rotor of ATP synthase, causing it to spin in a way that catalyzes phosphorylation of ADP to ADP, or ADP to ATP, adenosine diphosphate to triphosphate. This is an example of chemiosmosis, the use of energy in a proton gradient to drive cellular work. Fascinating that we have molecular machinery that acts in this way. This is just insane. Certain electron carriers in the electron transport chain accept and release protons along with electrons. In this way, the energy stored in the proton gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis. The proton gradient is referred to as a proton motive force, emphasizing its capacity to do work. In a similar way, in physics, electromotive force, or EMF, is called a voltage and involves electrons, but here we're looking at proton motive force. All right, we have our complex series of transferring our electrons, originally from our food molecules within this transport chain, helping pump proteins out into the intermembrane space, causing a huge gradient that, by chemiosmosis, forces hydrogen ions to go through ATP synthase back into the matrix, but at the same time providing energy that can phosphorylate diphosphate into adenosine triphosphate. Thus, we have the electron transport chain and chemiosmosis that form oxidative phosphorylation. During cellular respiration, most energy flows in this sequence. Glucose to reduce nicotinamide, to the electron transport chain, to the proton motive force, to ATP. About 34% of the energy in a glucose molecule is transferred to ATP during cellular respiration, making about 32 ATP molecules. The rest of the energy is lost as heat. So that would be 66% of the energy that's naturally in glucose we do not actually get to keep. But we do keep our bodies warm. So, glycolysis always produces 2 ATP. Citric acid cycle always produces a net of 2 ATP. And then the oxidative phosphorylation gets around 26 to 28, yielding about 30 to 32 ATP per individual glucose molecule. What we're looking at is a figure of the electron shell. Span membranes. All right, so using our reduced coenzymes. Fair enough. Showing more of those that eventually are our electron carriers that dump off those electrons, becoming oxidized during the oxidative phosphorylation process. All right. There are 3 reasons why the number of ATP is not known exactly. Photophosphorylation and the redox reactions are not directly coupled. The ratio of NADH to ATP molecules is not a whole number per se. ATP yield varies depending on whether electrons are passed to NAD+ or FAD in the mitochondrial matrix. The proton motive force is also used to drive other kinds of work as well. Most cellular respiration depends on electronegative oxygen to pull electrons down the transport chain. Without oxygen, the electron transport chain will cease to operate. In that case, glycolysis couples with anaerobic respiration or fermentation to produce a small amount of ATP when the electron transport chain is not available, or when oxygen is not available. Anaerobic respiration uses an electron transport chain with a final electron acceptor other than oxygen, sometimes something with sulfur, like sulfate. Fermentation uses substrate-level phosphorylation instead of an electron transport chain to generate some ATP. Fermentation consists of glycolysis plus reactions that generate NAD+, which can be reused by glycolysis. 2 common types of alcohol fermentation and lactic acid fermentation. In alcohol fermentation, pyruvate is converted to ethanol in 2 steps. The first step releases carbon dioxide from pyruvate. The second step produces NAD+ and ethanol. Alcohol fermentation by yeast is used in brewing, winemaking, and baking. what we're seeing here is that we still have glycolysis occurring in either alcohol fermentation or lactic acid fermentation. It's really just the end product that we are left with that's different. Either ethanol, 2 molecules of ethanol, or 2 molecules of lactate. As well as, yes. In lactic acid fermentation, pyruvate is reduced by NADH, forming NAD+, and lactate as end products with no release in carbon dioxide. Lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt. Human muscles can use lactic acid fermentation to generate ATP quickly during strenuous exercise when O2 is scarce. Fermentation, though, is beneficial to microorganisms or unicellular organisms that do not have large ATP needs, but is not sufficient to produce enough for us at our large multicellular scale. All use glycolysis, whether it's fermentation with aerobic or anaerobic respiration, to yield a net 2 ATP, to oxidize glucose and harvest the chemical energy for food. In all three, NAD+ is the oxidizing agent that accepts electrons during glycolysis. The process has different mechanisms for oxidizing NADH to NAD+. In fermentation, an organic molecule like pyruvate or acetaldehyde acts as the final electron acceptor. In cellular respiration, electrons are transferred to the electron transport chain instead. Cellular respiration produces 32 ATP and most per glucose molecule, whereas fermentation produces 2 ATP per glucose molecule. Obligate anaerobes carry out fermentation in anaerobic respiration and cannot survive in oxygen. Yeast and many bacteria are facultative anaerobes, meaning that they can survive using either fermentation or cellular respiration. In facultative anaerobe, pyruvate is a fork in the metabolic road that leads to 2 alternate catabolic routes. Depending on the availability of O2, pyruvate can either go through anaerobic fermentation or aerobic cellular respiration, when both are available. Glycolysis is an ancient process. Early prokaryotes likely used glycolysis to produce ATP before O2 accumulated in the atmosphere, used in both cellular respiration and fermentation. It is the most widespread metabolic pathway on Earth. The pathway occurs in the cytosol, so does not require the membrane-bound organelles in eukaryotic cells. Glycolysis and the citric acid cycle are major interactions to various catabolic and anabolic pathways. Catabolic pathways funnel electrons from many kinds of organic molecules into cellular respiration. Glycolysis accepts a wide range of carbohydrates, including starch, glycogen, and several disaccharides. Protein that are used for fuel must be digested to amino acids, and their amino groups must be removed. Fats are digested to glycerol, used to produce compounds needed for glycolysis, and fatty acids. Fatty acids are broken down by beta oxidation to yield acetyl-CoA, coenzyme A, NADH, and fat H2. An oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate. Something that should be noted is that even though fats can produce about twice as much ATP as burning carbohydrates, it takes more energy to get that process to occur. So looking at proteins, carbs, and fats, in their monomers of amino acids, sugars, or glycerols and fatty acids, our biochemistry has evolved such that all these can be used as starting points, though carbohydrates are usually the best. Carbs can enter in as glucose. Glycerols from broken down fats can enter in as glyceraldehyde 3-phosphate. Amino acids can enter in by the loss of an amino group to form pyruvate, effectively skipping part of glycolysis. Eventually, all these, the fatty acids or more amino acids, can be used eventually to form acetyl coenzyme A to power the citric acid cycle if we're not using carbohydrates. The body uses small molecules from food to build other than their own molecules, such as protein, other of their own molecules like proteins. These small molecules may come directly from food through glycolysis, or from the citric acid cycle. Feedback inhibition is the most common mechanism for metabolic control. If ATP concentration begins to drop, respiration speeds up when there is plenty of ATP. Respiration will then slow down when there's a lot. Control of catabolism is based mainly on regulating the activity of enzymes at strategic points in the catabolic pathway. One of the enzymes needed for glycolysis to occur is phosphofructokinase. Adenosine monophosphate stimulates the phosphofructokinase enzyme to occur in the forward direction toward pyruvate production. However, later on, an increase in adenosine triphosphate can inhibit phosphofructokinase, or the increase in concentration of citrate during the citric acid cycle can also inhibit phosphofructokinase, acting as signals that glucose doesn't need to be broken down and resources invested, while resources can be focused further down cellular respiration. Data from the experiments. This is just an example of thyroid hormone level and then oxygen consumption rate in nanomolars of molecular oxygen per minute milligram of cell. And it seems that oxygen is consumed at greater levels as the thyroid hormone level increases. The input of glucose to start glycolysis outputs 2 pyruvates, 2 initial ADPs net-wise, and then 2 NADHs. 2 pyruvates can be oxidized to 2 acetyl coenzyme A's, which bind to 2 oxalacetates in the citric acid cycle to output 2 ATP, 8 NADH, 2 fat H's, and 6 carbon dioxides. And then each of those reduced molecules can help input in electrons to release protons in the proton pump to eventually form multiple ATP. And this is just a beautiful representation of the intensive protein that we're looking at. This is just a biochemical enzymatic graph of phosphofructokinase enzyme activity. When there's a low ATP concentration, it is much more active, whereas when there's high ATP concentration, it is not as active with respect to fructose 6-phosphate concentration. Although eventually when there's a high enough fructose 6-phosphate concentration, activity seems to equal each other regardless of ATP concentration. pH difference across membranes with time. CoQ10, coenzyme Q, that can be used to help in times of lower energy. It's a useful coenzyme that's part of the electron transport chain and providing it to your body. sometimes can help you burn food more efficiently. In any case, this concludes Chapter 9 of Campbell Biology. Pleasure to have you all. So slightly past the 45-minute mark. Wishing you all peace.